Good afternoon. My name is Jim Simons, and it's my pleasure to welcome you to our virtual convocation. I want to begin by acknowledging and expressing gratitude for the fact that our country has just had a peaceful transition to a new administration, and it's something that we can never take for granted. And it's at the very heart of our democracy. And it's something that I want to acknowledge, as I say, and give thanks for. So our program today came about because my wife, Patty, who is sitting next to me here, is in a book club with Liz Harrington. And Liz Harrington is a famous English teacher and has had a wide influence here in California.
According to me.
According to Patty. And Liz happens to be married to a famous brain research scientist named Mike Harrington. And Patty and I got a chance to visit Mike at his office, at his building, where he has a very sophisticated, large research institute. So that's our program for today. And Patty will begin with our scripture.
Okay, I want to share Ecclesiastes 3, verses 1 through 9. And it begins with, there is an opportune time to do things, a right time for everything on the earth. There's a right time for birth and another for death, a right time to plant and another to reap, a right time to kill and another to heal, a right time to destroy and another to construct, a right time to cry and another to laugh, a right time to lament and another to cheer, a right time to make love and another to abstain, a right time to embrace and another to part, a right time to search and another to count your losses, a right time to hold on and another to let go, a right time to rip out and another to mend, a right time to shut up and another to speak up, a right time to love and another to hate, a right time to wage war and another to make peace. And I'd like to add my own, a right time to care about your brain, just for today.
And now it's a right time to pray. So let us join together in prayer. Oh God, we give thanks for a peaceful transition in our democracy. We give thanks for health workers in the midst of the pandemic, specifically for vaccinations here at Monte Vista Grove, which we've been allowed to have in the last week. Today, we give thanks for our brains, for a chance to think about how we think. And we're grateful for Mike Harrington, our speaker, and for his research that can lead to healing when our brains don't function like they should. So we pray for healing in our country, pray for healing among those who are stricken with the COVID, pray for healing of our aging brains. And again, we give thanks for Mike and his work. We pray in Jesus' name. Amen. Mike Harrington received his medical degree in 1976 from Glasgow University, Scotland, after which he trained in internal medicine and then neurology. He became a member of the Royal College of Physicians and Surgeons in the UK in 1979 and was elected fellow in 1993. Dr. Harrington spent 6 years in research at the National Institutes of Health in Bethesda, Maryland, followed by 9 years at Caltech here in Pasadena as a member of the Beckman Institute. He joined Huntington Medical Research Institutes and established its molecular neurology program in 1998, where he is now scientific director of Neurosciences. His objective is to find the cause of common brain disorders, none of which are currently known. He focuses on migraine and Alzheimer's disease, which share the same organ, but are not otherwise related. And so it's indeed a pleasure for me to welcome and introduce Mike Harrington.
Thank you. Thank you so much, Jim, and good afternoon, ladies and gentlemen. It's really a pleasure to have a chance to share some of the challenges and some of the excitement of the possibilities about our aging brains. And Jim is correct. Obviously, I had suggested, I wrote this, but we don't know the cause of any brain disease. Can you believe that? I mean, you probably think we in medicine know a lot more, but we actually don't know the cause of very many diseases. We don't know the cause of any brain disease, whether it's Alzheimer's or Parkinson's or multiple sclerosis or migraine. I call it migraine. If I come back to that, you'll just, I'll hopefully persuade Jim to pronounce it correctly. The world's becoming globalized, and that means generally Americanized. So even in Scotland now, they sometimes call it migraine. But I'll point out that the Greek origin, which is hemicrania, was the beginning of that word. And in Spanish, and I know you had a diversity experience recently, and we have rather a lot of Spanish-speaking people in our country, certainly in our state, and their word is migraña. So I like to call it migraine. What I'd like to do is share some images, and I'm just going to share my screen now to pull these up. Do you see these like purpley pink words?
Yeah, we got it.
You got it, good, good. Okay, so I'm just going to talk about our brains, Alzheimer's disease, and some new approaches. So what I hope to cover is an orientation to our brain. I don't suggest that you're not knowledgeable about our brains, but there's an awful lot that has been found, and I'd like to describe that to you. There's some really interesting things. I'd like to briefly tell you about current theories and treatments in this one sentence, because they don't solve the problem. We don't have any treatment that is effective. I'd like to therefore tell you what our research plan is, and then open it up to discussion. So I don't want to talk too long, but I do think there's some information that might be of interest to you. Why is this important? And I kind of love this. I adapted this slide, but it's called the 7 Ages of Dandelion. And I know when I was a kid, I used to like blowing dandelions to try and tell the time. That's what I was told I could do. But you start over here with a little sprig, and you grow up with this very fine bush at this stage. I've lost most of mine. But you can see what happens with age. And I think it portrays what's happening in our brain aging rather well. Age is, without question, the biggest risk factor for dementia, for many other things, as maybe some of you have found out. I know I'm not able to advance this. That's strange. Let me try down here. Okay. Why is this important? So I'd like to give a case history. Happens to be my grandmother, who I worshipped. So if you look at the picture on the left, the photograph she gave me, that's when she was 19. And you can, beauty was not passed on to the males of our family, but I think she was stunning. I never saw her beyond the photograph at that stage. And the photograph on the right is when she was 20. She was in South Africa, in Port Elizabeth, in 1906. And that's her. She was a concert pianist. She has this kind of bouffant dress, and she's playing the piano. In fact, what she told me, she was playing something from Chaminade, who I'd never heard of, and at the time, which is terrible. So Chaminade was this 19th century female composer. And at that stage, I hadn't known there were any female piano composers, classical music composers. So my grandmother, Sybil, was way ahead of the curve, certainly way ahead of me, always. After she had a breeding program that produced my father, among other offspring. And after she dealt with that, by this time, I should say, she'd met the man who became my grandfather and had that breeding program. But then once they were out of the house, she started painting, and she was exhibiting nationally in 1956. But in 1967, her memory and a lot of other things deteriorated. What I remember was when I used to visit her, this is in Glasgow, when I was leaving, we'd both hold each other's, we were both right-handed, hold each other's hands, put our left hand on the shoulder, and we'd kiss each other goodbye. And when I went to do this, she stubbed a cigarette out in the palm of my hand. She'd never smoked in her life before. When I look back, this was the beginning of her dementia. And I remember leaving, my mother said to my father on the way out, "You have to do something about that woman." And I rather cheekily said, "That woman is my grandmother." And she deteriorated sort of over 10 years and got to the stage where, you know, I'd go in, sometimes she would know who I was. 5 minutes later, she wouldn't know who I was, or she'd say, "I know I'm supposed to know who you are. What's your name?" And I'm sure you all have friends, family, relatives who've succumbed to dementia. It's just terrible. You know, it robs us of the person. It robs that person and their loved ones. So this is terrible. And to put it in broad context, here's a plot of the prevalence, the number, the frequency, we call it prevalence in sort of epidemiological speak, of common brain disorders. And you can see number one in terms of frequency is migraine. If I was with you, I would have you all pronounce it, but that's not very good in a virtual program. 12.1% of the population. Alzheimer's is next. And then these are more than all of the other neurological diseases in terms of prevalence added together. So they're big problems. And these are the two we're studying because we don't know the cause of them. I'm a simple person in terms of the way I approach our science. How can you treat something if you don't know what you're treating? And that's just, to this day, to me, stunning that we don't know the cause of these disorders. So I'd love to speak to you at another time if you're interested in migraine, but it's going to be an Alzheimer's today. And if we look at this plot on the right, all over the world, and that's what's displayed here in different colors, as you get older, the prevalence rises, the incidence rises. And it's the same all over the world. It's actually, the curve is a little pushed over to the right, mostly in India. I don't know. I think this is maybe why people tried trials of turmeric. I know at UCLA they had a trial that was somewhat positive. I personally think, how could you pick out turmeric when there's a lot of other different things in the Indians? But their slope is the same all over the world, north, south, east, or west. Age is the biggest risk factor. I'm trying to advance. Okay, so there's no current treatment I've just mentioned to correct it, and the best advice currently is to reduce dementia risk factors. So I'm not trying to be depressive towards you, but there are things we can do. And the first thing is you have to go out and pick your parents better, or not if you've got good parents. So genetically, there is a risk. What is the risk? In the early onset, which I'm not going to discuss, which is very rare and it's nasty, there are mutations. If you get one of those, it's not a risk. It's a guarantee of Alzheimer's. So the more common thing that we're all worried about, we call it late onset Alzheimer's. It's more common in families who have parents with a history of it. Often, you know, I don't know about you, but when I ask my parents, when I was come over to the States, they were visiting from Scotland. I was doing a lot of lab work at NIH in Bethesda, and at dinner I sort of asked my, at a reasonably appropriate time in the conversation, I thought, you know, what are the illnesses that run in our family? And my father quickly replied in front of my 6 and 4-year-old son, "It's none of your effing business." And this was not an uncommon attitude back in the day. You know, you wouldn't talk, you wouldn't disclose, you wouldn't show your weakness or something. But if you do know your family's, your ancestors' cause of death, and there's no dementia in the family versus a family that does have a dementia, it's very unclear. We don't know. But the epidemiologists have said you've got about a 4-fold higher risk if there's family illness. And there are 29 known genes that are involved. ApoE is the only one that's really a common one. And but in most people's situations, we just don't know. If there's a history in your family, you've got a slightly increased risk. I'm joking about the fact that you can do something about that. It's a little late. They made the decision for us. This is very interesting. So good exercise, good education, both, and the exercise, cognitive and physical exercise, and enjoying good social relations, all of these have been shown in very high quality studies to be protective. And I'll just point out, you know, good education is something we do when we're young. So a behavior of when we're young is protective in every study that's ever been published. I don't know an exception. It's protective against a disorder that comes on when we're old. And go figure, isn't that interesting? What is the resilience factor? It's not known. It's a understudy, but we really don't know. It's really curious. Not something we can go back and do, but the exercise, so if you've got bad arthritis or other conditions, which are common aging conditions, cognitive exercise, as I joke, use it or lose it, is a protective thing. The good social relationships, the partnerships that people have, these are proven to have protective effect. Avoid brain trauma. Why they allow boxing, pugilism to continue beats the heck out of me. I volunteered when I was asked to be the doc for the Scottish boxing team when I was still working over there, and I took the job on, but I only lasted a month because the first punch that was landed, I stopped the fight. You could pick up changes in the brain function. I'm sure you all, this makes common sense, which is surprisingly uncommon. But the pugilism is obviously terrible, but the NFL, do you let your kids, your grandkids play soccer and head the ball? These are all serious issues because the evidence is just far too inadequate, but it's accumulating that brain trauma, particularly repetitive brain trauma, is damaging. Smoking, huge risk factor for dementia, which many people don't know. And then treat the conditions. This is something we can do that we know damage the brain. And the common ones are really common: high blood sugar, high blood pressure, high blood cholesterol. And there's good evidence, not as nice or tidy as we'd like, but there is good evidence that if these are treated, you reduce the increased risk that untreated conditions would confer on you. So those are really, you know, what I like about this is it gives you a reason to take a daily medication. Who wants to take something daily when you don't even have symptoms? And you know there's side effects of these drugs. These are the good reasons to take these prophylactic medicine because they reduce the risk of the damage. I want to show you some data that's, I think, interesting. Here is brain volume plotted by age and sex. So we got, if you look at the top plot first, and I think typically I chose blue for the males, red for the females. And you can see a couple of things. We've got bigger brains than the women. And I'll immediately destroy that, because when you normalize for body weight, body mass index, the women have bigger brains than us. So that's destroyed any size matters bragging by the males. Red, what you see though over time is the brain gets smaller with age. And this is not Alzheimer's, this is normal aging. So it's pretty big relative to size when we're born, but as you can see down here, but it clearly gets smaller with age. And I won't ask for you to put your hands up because I'm not looking at the images just now as to who has a little problem as they get older, recalling a word. I know I've always had that problem. And we don't have a good way of distinguishing this, but with experience in the field, we're not too bad at distinguishing what we call typical aging, which is a consequence, I'm pretty sure, of this smaller brain. We do have reduced ability to multitask. I've never been good at multitasking. I'm even worse now than when I was younger. And I'm still really young. I thought I'd show this. Is really interesting about our brains. So this is relative size. So size matters, yes and no, but it's just interesting. This lady's the honcho, the real guru of doing multi-species studies, particularly for the brain. If you look, here's well, let me go first to the human brain here, top right at about 1, 2 o'clock. You can see that these are relative sizes of the brain in many mammals. The biggest one is the African elephant. You know, one could joke about their lack of forgetfulness. Here's a dolphin down here, and this is a zebra over here. We can see some pretty sharp animals. Remember the mice and the rodents? Here's a rat, and I'm blocking where the mouse is just now. It's in there somewhere. Their genome is not that different from ours, but you can see their brain's a lot smaller. They clearly use it very, very well. I just thought this is interesting for size, because I'm going to come back to that in a minute. Now, the scale of the problem of our ignorance, trying to figure out what's going on, is that the brain is not the only organ involved in Alzheimer's disease. Data is not very extensive on this. We're studying it a lot. But neurons, the nerve cells of our body, I don't know if you knew this, don't divide again. They're what we call post-mitotic. So when you're an infant, you've got your lot. There's a few stem cells, but they're a drop in the ocean, just to be simplifying it. So over the first 20 years of our life, things organize, get pruned and sorted out. But from there on, as we all know, 20 years of age, life is downhill from there in many respects. And but the neurons, which don't divide again, they remodel. Imagine if you have a car that you ran for 70 years, every millisecond of every day for 70 years, no mechanic would ever keep it running. So our neurons do pretty darn well. They're remodeled, the components are replaced, but they're not dividing again. They're not starting afresh. And that's pretty unique for the neurons. And they innovate all of these structures. And I think with time, all of them will be known to be affected by Alzheimer's disease. It's just the main symptoms are not there. The main symptoms, as we know, are in our cognition or lack thereof. If you look on the right here, the central nervous system cartoon of the peripheral nervous system, we've got 45 miles of nerves in the skin. There'll be a quiz afterwards, so I'm just warning you. You have to remember these things. So there's a lot that can go wrong. If we switch to what I call the molecular level, the problem's way bigger there. The challenge is way bigger. And again, to me, this is very exciting that the challenge is so immense. It means we should be able to make huge differences, huge progress. Huge is a word I've rarely used for the last 4 years because our POTUS used it so frequently and abused it so frequently. Anyway, each person is different. That's, I think we all know. Each neuron is different, each nerve cell. And we've got the more recent estimate is about 86 billion. Of course, no one's counted that. That's in our brain. It used to be referred to as 100 billion neurons. That's a lot of cells, and every one of those is different. I remember this number when I was younger because for fruit, the most abundantly consumed fruit on the planet Earth are bananas. And I read just a few years ago in National Geographic that about the different commonly eaten fruit. There are 100 billion bananas eaten on the planet every year. Well, we have about that number of neurons, and they're all different. And there's a lot of other cells in the brain, and each cell is continually changing composition. Every millisecond, that's proteins, lipids, small molecules, big molecules. Otherwise, you're what I call dead. And so this is immense. We don't know the composition of any cell, any tissue, or any fluid. We know quite a lot of some of the components of some, but we don't know what any of our cells are made of, let alone how they change with what I call clinical variation, you know, the time of the day, our age, our gender, our physiological variation, disease, and so forth. So I think you get an idea of the immensity of our lack of knowledge, which is the source of why we don't understand our diseases or our health very well. I wanted to show you some images, well, just one here. And I want you just to focus on this right-hand image in a minute, because again, when I was really young being taught it, we were led to believe this organ, our brain, was like a solid, stuck, discrete region, like 1.2 kilograms, and it didn't change. And it's actually incredibly dynamic, not just the millisecond molecular changes I was talking to you about, electrical signaling and everything, but it's actually physically incredibly dynamic. At a molecular level, if I squirt physiological levels of estrogen into your arm, you'll improve your verbal memory, which is something I might benefit from. If I squirt physiological amounts of testosterone into your vein, you would improve your three-dimensional structure solving capability. Isn't that interesting? Physiological levels of these sex hormones affect your brain function. When I tell young ladies in my lab, they sort of say, yeah, we know. I'm not sure if we're as alert to this for many reasons as men, not just because we don't have such overt hormone changes. I just don't think we're as smart. But anyway, we don't live as long as women either. One of my colleagues in Amsterdam's the head of the Alzheimer's Center there, very, very renowned man. And he's got 200 cognitively healthy centenarians. There's no males. And he didn't try to avoid males. We just don't live as long as the women. And isn't that interesting? Anyway, so back to this image. I think this will show you how dynamic the brain is, because this is going to show you what the movement in our brains during the heartbeat, it looks like. Oh, yeah. So isn't that wild? So I don't know if you can feel your brain moving around like that. It's pretty dramatic. I know I feel if I get exercise, I feel I'm flushing my brain. I've always felt that. And I think that's probably actually happening. What's interesting in here, I study spinal fluid a lot. And I'd first, when I was in America on the East Coast, I'd actually discovered, and then later at Caltech, finally sequenced the protein that's the basis for the diagnostic test for mad cow disease. And Creutzfeldt-Jakob disease, it's called. And it's interesting. So I've studied spinal fluid a lot, and this dynamic fluid that you can see, the white region here that goes all the way around the brain, is changing, and it's moved primarily by our heartbeat, also by respiration, which is not really shown for. They've not shown it directly in this imaging sequence. And it changes day and night. And you've heard of beta amyloid, which I'm not going to be talking about too much. It's a major protein that's abnormal during in Alzheimer's disease in the brain. And it's deposited in the brain. I'm going to show you some data on this. The clearance of this goes through the spinal fluid, and the clearance increases at night when you're sleeping. And when this first came out in 2013, I was at a meeting when all the gurus were going around saying, I've got to get more sleep. I've got to get more sleep. I need my sleep to clear the amyloid. And so I just thought these are interesting updates on the brain. So to try and figure things out, learn more, maybe identify some targets that could help us treat Alzheimer's, stop it, delay it. We've got a research program in Pasadena. Love to recruit you or have you visit. And so basically what we do is we recruit and classify clinically cognitively healthy study participants. Some people think they're cognitively healthy and they're not. Some people think they are dementing and they're not. We do a very major assessment on quite a few visits, and then we have 10 to 20 experts. We do this twice a month. We had one on Saturday, and we present all the data for an individual clinically, and we don't stop until we all agree on the clinical classification. Are they cognitively healthy? Do they have mild cognitive impairment? Do they have dementia? If they do, is this of the Alzheimer type or maybe one of the other types of dementia? Because there are other causes. And the neuropathologists will basically say, you know, the clinicians don't know. When we have to chop up the brain and then we know what's going on there. Because clinically, we're not very good at it. We're not as bad as they would say, but it's complicated because as we get older, a lot of things happen. And Alzheimer's is probably very heterogeneous. Lots of different events going on as we get older. You know, I probably find there's not many of you in the audience who don't have multiple little things that are going wrong. I won't focus any more on that. So we classify people clinically based on, you don't need to pay attention to this if it's not your thing, but there's national criteria. So our study participants go into the national database. It's called the National Alzheimer's Consensus Consortium. And we have this consensus conference, which is the gold standard. And it's wonderful. We learn so much from each other because we have neuropsychologists, cardiologists, neurologists, etc. Young people, old people debating. And then we collect spinal fluid and we determine the presence of normal or abnormal amyloid and tau, which are known biomarkers of Alzheimer's. They're not the cause, I don't think, because of all the trials that have failed when directed at removing those proteins. But there's still efforts that'll go on till the year 2030, at least in the clinical trials that have been started to see if removing these bad proteins will help people. But it's a little negative at the moment. The way we can have a good biomarker, even if we don't think it's the cause, as I don't, but a biomarker of disease, we can either image it in the brain or we can measure it in the spinal fluid. We do both, but imaging it in the brain is pretty expensive. It's about $3,000 a pop, and we've got research money to do it, but it's not done very much for obvious reasons. We have to infuse a radio-labeled ligand that'll attach to amyloid, and we have a PET scanner that'll This is at USC, not at HMRI, but our study participants go there and we can look at that. Spinal fluid, you know, who wants to have a lumbar puncture? I have half a dozen, and I'm not a masochist. By people who are good at it, it's a very straightforward procedure, and it's just a lot simpler. And we study molecules in there, in the blood, in the urine, that I'm going to give you some indications of progress with. We do brain imaging. That's what the magnetic resonance imaging stands for. We examine the heart. We're doing a lot of what we call brain-heart work. I like my very famous cardiologist who works in our institute, Bob Cloner, who at a meeting today was, when I was poking fun at him, he said, yes, the heart's only purpose is to supply blood and nutrients to the brain. But if you don't have good stuff going up into the brain, yeah, it's not going to work. We do EEG, we do retinal studies, and I'll give you some indication of progress with these. And we follow this stands for study participants longitudinally to evaluate the predictive value and to search for mechanisms from all of the early biomarkers. I realize with time I'm going to have to be quick. So we can detect people who are cognitively healthy who have bad biomarkers. And this is the way we do it. The red is all the Alzheimer's. There's some Alzheimer's, 4 of them, who have normal values. The blue are cognitively healthy, indistinguishable from each other, but all of these guys here are at higher risk. And when we follow them over time, they decline cognitively at a faster, much faster rate than the ones on the other side of the line. Here's the healthy brain on the left, age-matched severe Alzheimer's. Isn't that amazing? You lose hundreds of grams of this organ with Alzheimer's. Where does it go? And my joke has always been, you whiz it away. Where else would it go? It's not going to come out of your ear or your nose. And so we decided to look at urine to see if we could see evidence of this. And we just published this recently, where in cognitively healthy people with normal chemistry, they have high levels of this energy molecules. These are dicarboxylic acids. We have low levels of these other DCAs, which are chemically broken down fatty acids. Alzheimer's is quite the opposite. They've lost some energy molecules and they've increased their chemically cleaved fatty acids. And the guys who are cognitively healthy who've got bad chemistry are in between. So we're very excited about this. We think this would be a lovely screen test. So I do a lot of lumbar punctures for spinal fluid, but you know, that's not something for the world's screening. A urine test, everyone can supply that body fluid. Not everyone can have a lumbar puncture or an MRI scan. So this is very exciting. The values of these compounds match or correlate with the shrinking size of the hippocampus, which is suggestive that this urine measure is primarily coming from the brain. Blood lipid changes. Here are images of PET scanning where normal Alzheimer's, you can see a lot of PET binding. And we've shown recently that the fatty acids in the blood are actually altered in the people with Alzheimer's compared to controls. We can do a lot of electrophysiological tests, and that's what's displayed here. You can give a signal, look at a peak, see if it changes. You can have someone, I don't know if any of us had a treadmill test, cardiac risk for coronary disease. Your EKG is normal. You go on a treadmill with the electrodes, and if the STT segments go up, you go to the cath lab and your life gets saved with a stent or cabbage or something. I want the same for the brain, but you don't go on a treadmill. You, like this lady, you turn around and look at the computer and do simple tests. And Siang Hong Arakaki, my lab's doing fantastic work there with cognitive challenge instead of the treadmill challenge. And in our cognitively healthy people, she can pick up that the brain, even though they get the correct answer, the brain is seen to be working harder to get the same correct answer. And these are other tools that we use. Here is a nice one where we're testing sensory motor. If you look on the left, this is a path. It's like an obstacle course. You've got to, on the wheel on the right, the subject, and she's very old, as you can see, has to negotiate this path. And we can measure the error or the lack of error. And so this is, she's got to be able to turn the wheel. She's got to see the screen here. And then we add complications, like we have the chair bouncing or the wheel bounces. So then she has, it's like going on a mountain bike. So we have our study participants doing this. The eye, I mentioned, this technology, I don't know if any of you have had an OCT, optical coherence tomography. It's so cool. And it is, the retina is the eye into the brain. So Alfredo, our amazing ophthalmology colleague, 30 years ago showed that the retinal nerve cells are lost in Alzheimer's. But this was, you know, terminal Alzheimer's, whose eyeballs had been donated along with their brains. I wanted to see in our early cognitively healthy people, did they lose neurons? And using this technology, measuring the nerves in different places, this is the normal chemistry, this is the bad chemistry. So they've got thinner retina. So we could imagine, this is a couple of minutes to take a fancy camera image of your eye. I'm gonna rapidly go through some more data. In spinal fluid, I mentioned we've studied a lot. And we were the first to find there are a lot of particles in there which had never been found before. We published it in 2009. There are in a healthy people, a healthy person like myself, there are 4 billion of these per milliliter. In Alzheimer's, they go up to 7 billion. And these are very interesting. They contain neurotransmitters, they contain enzymes, they have, I could talk a while on describing these. It'll take thousands of years to figure out what they all do. But they give us a source of brain-derived membranes. Here's a large dense core vesicle. You can see the membrane. We can measure the thickness. We can measure the chemistry of it. And we see changes that are informing us of early brain membrane defects in Alzheimer's. The last thing I want to show you, I'm very excited. We were able to persuade the Keck Foundation, the WM Keck Foundation, and so it's a very prestigious award they gave us because from my migraine work I found sodium is very central in its fluctuations in causing migraine. And we've discovered how to do this and measure this in humans. I've been doing it in rats for a long time. So here's a normal proton MRI. Here's the same person, sodium. So all you're looking at is basically water. It's the proton. Here, all you're looking at is sodium. And you can see the ventricles and everything. What I pitched to them was that fluctuations in brain function arise from fluctuations in sodium in the brain. And we've shown this in migraine. We're now proposing that when my grandmother 1 minute knows who I am, the next minute hasn't a clue who I am. I don't think her pathology is changing at that time. I think it's her sodium. So we're in the middle of testing that. I'd like to stop now, but I think I hope I've given you a sense of, there's a lot of excitement about what we should be able to find in our brains. I'm realistic. It's not going to be in time for me, maybe not even our sons. We have 3 sons, but maybe the most beautiful girl in the world and the most handsome boy in the world, my grandkids, maybe we'll know better for them, because it's predicted by 2050, if we don't slow this down, the planet will be bankrupt. We will not be able to manage with the increased longevity, lifespan of people. We'll not be able to cope with it. So I am excited for them. I'm a little bit more realistic for myself. So at that stage, I'm going to stop because I've gone on longer than I thought. And I hope there's some time for questions. Jim, would there be time? Or Lindsay, would there be time?
Absolutely. There is 15 minutes for questions. And Mike, I'd like to start with one. I was struck by, you're talking about boxers, you know, they're just looking for trouble. The soccer players, my grandson, when he started playing soccer at the age of 5, they weren't allowed to do headshots. But give another 4 or 5 years, now he's older and he is doing headshots. And what you're saying is it's utterly stupid. We're just looking for trouble. Is there any way to, I guess this is a political question, is there any way to change that for, is anybody, who's working on that?
Yeah, it's, well, gosh, it's a long, slow, changes happen in spurts. The people are working on it more than before, but there are big problems. So I think political, yes, but socially, culturally, it's a huge problem. Because, you know, the macho stuff, and it's for women as well as men, but, you know, you're in the sport, you want to go back in, especially for the kids. And one concussion is not good. Repeated concussions increase the risk of brain damage. We call it CTE, or chronic traumatic encephalopathy. They increase the risk a lot. And if you have that one fairly quickly, the second one fairly quickly after the first, it's bad. And so there's some ground rules, often totally ignored. You know how reluctant the NFL was to acknowledge the problem. They've acknowledged it now, but you know, it's tough. That's a multi-billion-dollar industry. Do you think they're going to stop the way they make money just now? Actually, COVID, I guess, has stopped a lot of that. It's a real problem, Jim. And the data from the studies on soccer, the best one was they took people who headed the ball less than 3,000 times a year. That was the low. This is, you know, professional players, or more than 3,000. And they studied a lot of them. And the cognition was poorer in the ones who headed the ball more. And at a substantive, significant, measurable level. The other part of the problem, society, cultures, and our ignorance is a problem. We don't have a good way of testing it. So we had a big Army-funded study at Huntington Hospital, concussion. So head injuries that go there, they CAT scan. If the CAT scan shows no trauma, they kick them out, usually giving them narcotics. So it's terrible, but some of my colleagues work there. But that's the knee-jerk response. Not good treatment at all. But we got exposed to those people. They were given the chance when I did my Spanish Inquisition on them to join our study. And we were looking for biomarkers. And we did things that I just showed you about with the EEG, with blood biomarkers, and we didn't take spinal fluid. The first guy I saw was a trauma-controlled non-head injury. That's how he was referred to me. This is a 20-year-old Marine. And he's talking very slowly to me. And he'd been out of Camp Pendleton on the freeway, made a mistake in the LA freeways, right? He has a car crash and he's got abrasions down his leg. And I'm realizing he's not normal. He's not a 20-year-old Marine. And I talked to him a little bit and he's clearly had a concussion, but he doesn't know because his bell was rung. The docs don't know because they don't have a test for it. I know just because I'm interested in it. So 2 weeks later, we saw him. I asked him how he felt. Oh, I feel as if I'm in a dream. This is a 20-year-old Marine. A month after the injury, I said, how do you feel now? He said, I feel great. I'm back. So in other words, this guy clearly had a significant brain dysfunction for nearly a month. Not diagnosed. I mean, how common is that? It's, you know, probably all boys, we probably all had these concussions. The amazing thing, our bodies are resilient, so we can accommodate some of it, but not too much. If you have an ApoE4 genotype, you're more at risk for Alzheimer's. You're also more at risk for chronic traumatic encephalopathy. So, but we don't have good tests. And, you know, there's people, our institute tried to have the NFL or the colleges that play at our, well, UCLA, who plays at our Rose Bowl, have an MRI down there so we could scan people. There's efforts, and eventually, slowly, I believe we'll learn more. But I would plead with you, I'm not trying to persuade you, we have to participate in research. We have to be more proactive, because without knowledge, we can't do anything, right? We don't have our fantastic Zoom capabilities and technologies without people making advances. And yeah, and we don't, I'll tell you another thing, we talked about earlier about diversity. Do you know, we have African Americans in our study, not many. My study is local, it's not population appropriate, it's mainly white females. Why? I found out all the Alzheimer's studies in America pretty well have more females. And I was obsessed with this, and no one really knows. I think women are just more altruistic than men. So my typical retired couple, the woman wants to do the study, I asked the husband, well, I'm busy, you know, this octogenarian who's retired, I'm busy. And we have some, it's about 64% females. I'm actively recruiting African Americans. Do you know why? African Americans have twice as much Alzheimer's than white guys. This is huge. We don't know why. And I could talk more about that, but there's a lot we need to do for concussion. Sorry, that's a long ramble to your question, Jim. I'm taking the time from other questions.
I have a question.
Yeah.
This is Helen Bott. Not so much a question. Hi, Mike.
Hi, Helen. It's lovely to see you.
It's very nice to see you. I've had the privilege of being on the board of HMRI for the last 6 years, and I'm entering my third term, 7 years. And I'd like to put in a plug to each of us to consider being part of Mike's research program. It really will give us invaluable information as time goes on. My late husband, John, was honored to be part of it and felt it was almost a duty of his to be part of it. So thank you, Mike, and thank you all the researchers at HMRI for the wonderful work that you're doing.
No, thank you, Helen. I mean, John, your late husband, was amazing, and you probably wouldn't imagine that he was cognitively healthy, but he was very cognitively healthy.
Yes, I know. Some days too much so.
And the other thing, our consent form.
A little so they'll see it.
Hello.
Say again.
No, who else is asking? I have a question, but it really relates to working with students. And when I was teaching summer school, oftentimes every student in the class, which was science, medicine, and biotech, wanted to help their grandparents. And so we talked a lot about the kinds of things they could do. And many of them said, well, I could teach my grandparents how to play games on the cell phone. And I said, well, if you do that, teach them how to play the same game, but give them an iPad, because the size is too small on your cell phone. And then they would come up with other kinds of things. And they really were concerned. So if you do the requesting help, and you tell the grandparents, the older ones, that if they lend themselves to your program, their grandkids are going to be grateful, because they were all, these 7th grade kids and going into 8th grade, were all really, really concerned about the health of their grandparents. And my other advice to them was, be nice to your mom, volunteer to do the dishes, because your mom is even more concerned than you are. And they would come back and say, my mom tells you thank you. So I know that that's a really important part of the communication, is the family dynamics. And then just yesterday, I went to visit a student who's graduated from Stanford and is now going to Caltech. And he's doing something new with his research. He's in medical genetics. And he's working on the cell. He's not working with genes necessarily, but he's trying to do open cell medical chemistry. And he gave credit to Francis Arnold from Caltech in protein chemistry. And I wondered if you are sharing any of your data with migraine studies with other people at Caltech that are working on cell protein chemistry and lipid chemistry.
Oh, thank you, Patty. Yeah, we've bumped into each other a lot over the years, and you're one of the more infamous and highly regarded teachers in this whole area. And but to your question, yeah, we've not enough, but it's, you know, bandwidth. How many things can you do well?
Yeah, okay.
But yeah, I've got two postdocs that are in Caltech labs just now. They're both in chemistry, not biology. So Bob Grubbs, Nobel laureate in chemistry, has got JK is a, well, Korean originally, but he did his PhD at UCLA, and he's now in my program, but he's working in Bob's lab. And we just had a lab meeting at lunch at 12 to 1 today. He was giving us an update. He's, the urine measure I told you about briefly is not ready for prime time. Technically, you wouldn't want to do this. It's a lot of chemistry. And I want it simplified, right? The sample is simple. I want the chemistry. So JK simplified this, and it's looking very close to us testing on our human urine samples. And then Veronica Hubble, which is a great Caltech name. She's not related to the telescope at all. She's somewhere in the South, one of these southern states in America. She's got a very interesting accent. She's working in Brian Stoltz's lab, and she's synthesizing new molecules to regulate the sodium in the brain. And it's really exciting. And we've got an in silico chemist, biophysicist, who is in a private company now. He left Amgen, and he's working with Brian Stoltz's lab.
On a break.
Sorry?
You know,
I think they're just waiting for the next question, but go ahead, Michael. And sorry, while I'm interrupting you anyway, I just wanted to also call your attention to, we have some messages in the chat box when you get to them.
Yeah.
And just for you and the audience, I know we're right about at time. If Michael's willing to stay a couple minutes, I mean, there's no rush, but just so you know, get your questions out.
Well, thank you, Lindsay. So I'll read these questions. Is AD the most common cause of dementia? What is the prevalence of vascular dementia? Very good question. It's a mix. The older we get, the more of a mix. So it's believed over 80, it's more often, the most common form is mixed dementia. Under 80, the most common is Alzheimer's. The other ones that are not too uncommon, we've got a few of each of these. Lewy body disease, my hero comedian Robin Williams, who died of that. We have, I didn't have him in my study. We have several people with Lewy body disease, very common with Parkinson's disease as a late consequence of that pathology. Frontotemporal dementia, where is either a language or a behavioral prominent presentation, and vascular dementia. And the vascular, lots of fairly big strokes or dementias, but the one that's most common is microvascular, and it's where a lot of the cabling connections in the white matter are disrupted. And actually, Helena Xu at USC is probably one of the world, well, definitely one of the world experts in that, and she collaborates extensively with this. Creutzfeldt-Jakob disease I mentioned earlier can cause dementia. It's much less common. Normal pressure hydrocephalus is actually something we can treat, or the neurosurgeons can treat, and one of our participants had that. And it presents, they all present a little differently. Normal pressure hydrocephalus is really interesting, Barbara, because it's, there is a treatment. It's it's a very bad name for it. We don't have a better name. But if you shunt these people, they get better. Not always, but the triad of presentation is poor memory, poor cognition, I should say, poor balance, and poor urine control. And yeah, so I hope that answers your question, Barbara. Larry Ballinger asks, would I correctly assume that volunteers are sought around LA for studies? Great question. A lady journalist in Long Beach wrote an article. I get calls. I don't want to put people off, but it's awkward. They have to schlep over from Long Beach. That's not good. They're now, several of them well into their 90s, and we Uber them over. You know, it's so yeah, we prefer as local as possible just for transport, because they have ideally up to 10 visits in a full assessment. And we do that every 2 years. And if they want, when they stop using their brain, which is my euphemism for they die, they can give us their brains. And we've got 16 of their brains after they stopped using them. I have a husband and wife whose brains are side by side in the freezer, which is what they wanted. It's very romantic. And then Helena, they were going to throw out the brains from vascular dementia that she'd collected over 30 years at USC. Can you believe it? They were going to throw them out, and she studied them during life. I said, no. So I have 230 of her brains as well. So we're just trying to get funding to actually look at them properly. Betty Nelson asks, African Americans having more AD. Anyone studying the immense stress they tend to live under from their earliest years? Very good question. They're really very little. There's just to let you know, because we're not the only ones starting this, there's a group at Rush in Chicago, very top Alzheimer's research group, and one of their studies is exclusively on African Americans. So that's really good. And then Harvard has a good study on African Americans. And that's about it. You know, most of us have not been studying it. So I've told you, I'm aiming for 50-50 black, white, you know, so we've got an outreach committee of African-American experts in the community because, you know, for those of you who know ethics, you know, Tuskegee and all these things, they have every reason to be very suspicious of not going into studies. I worked with the lady sociologist at USC. I helped her with a study on African Americans, and I was the only white guy in the room on 2 occasions for a total of 200 African Americans. And she was trying to find out why they don't do studies. They don't participate in research or anything. And it's basically no one speaks their language. No one approaches them properly. So not a trivial thing to try and look at. And I agree with your question, Betty. We have to explore that because I don't know why they have more of Alzheimer's. Libby, tell us about sodium impact. Did your grandmother have to? So in migraine, I've not studied it yet in Alzheimer's. It's not been studied there. In migraine, we found it's very important. No one had studied the sodium. We did an epidemiological studies. My statistician never, ever, ever wants to see that database ever again. It's the National Nutritional Database. And the bottom line is migraineurs take less sodium in their diet than non-migraineurs. And I think, you know, and no one, of course, most of all us, we don't know why. Our body handles sodium different in our kidneys, from our salivary gland, when we're drooling over some nice food. I hope we're not, you know, moving fluid around in our other organs, so it's handled very exquisitely differently by our body. And yeah, we're going to look at firstly just measuring sodium. Sorry?
Michael, I'd just like to second what Helen said. This is Norm Thomas and Meg Gautier. We've been participating for 10 years in the study at Keck School of Medicine at USC, the longitudinal AGNI study, and we commend it. And there are so many studies going on now that they are seeking for recruits for, and I hope many at the Grove will consider it.
I think it's very important to do it. I mean, you got a brain, you can let them study it. And what makes it easy, it's only once a year that you go down there. So it's not a big trial.
Is that the one, because there's quite a lot there, is that the one at the Keck School or down?
Yes, at Keck School.
Okay, okay, yeah.
Right.
They're wonderful guys. We have grants in common with them there. It's a, there's this bad press with USC for things that we won't talk about. They're terrible things. But there's really good things there. And the guys working on dementia there are stellar. You know, Paul Azen was recruited up and his group from UC San Diego, and there's a lawsuit over it. His money comes into everything. He's the senior guru in charge of most of the, about $100 million or more worth of trials going on in America. I mean, they're a huge group, very important group. Betsa Lokovic is one of our main collaborators. Wonderful scientist and group, and Helena Shu as well, Lon Schneider, the huge list.
We've donated our brains in the program.
You've donated them already?
In our will, not before that.
Oh, Norman, I was worried for a minute. I thought you must have an implant or something.
I'd like to ask a question.
Yes, sir.
You've mentioned that you're doing studies on racial studies on white and black. What about Hispanic and Asian?
It's thank you very much, Dean. It's all bandwidth, so we can't. I took the one that's got the biggest difference. I'm really interested in African Americans because of this twofold increase. The Latinos, Latinxes do have a lot, but there's a huge study based in Dallas on Latinos, and USC have a big Latino study going on. And actually UC Davis has some African American study going on. I forgot about them. So we wanted to do something and we collaborate with USC a lot already, that was different, that would be additive more. But yeah, there are good studies for Latinos. I'm not aware of particularly big Asian studies going on. So, but that's, yeah, I don't know. It doesn't mean there aren't good Asian studies going on. I don't know everything. You probably learned that about me just now. There's so little. As you get older, you realize you know so little. You learn more, and it just proves that you know less. I don't know what that is. There's another question here that Lindsay's relaying. Will I say something about studies about vitamin D levels? Not a lot. Vitamin D's important. All vitamins are. It's not clear there's any great role for vitamins in our brain health. The studies that have followed vitamin supplementation through quite a number of vitamins, they're important. We need them for our bodies. There's no real strong reason that they're important in an inordinate way for brain health. And this is true across the board. I mean, the omega-3 fatty acids, which we're looking at very carefully, are important. We don't know why. We don't know how to get them into the brain. That's why we're doing studies. We're publishing pilot studies now, and our colleague at USC, Hussein Yassin, is the lead guy on this, where we don't know how a lot of the molecules get into our brain. So you'll have heard of the blood-brain barrier, and there's the blood-CSF barrier. Nothing gets into our brain without controlled passage. It doesn't just diffuse in. The rest of our body, it just sloshes around, goes through our blood supply, can go out into the tissues. And that's not in the brain. So that means you've got huge energetic processes moving every darn thing to get into the brain. It's remarkable how this is done, right? How do we get enough glucose, which is the main nutrient for our brain? You know, ATP is consumed big time in moving it across through the glutamate, the glucose transporter, and so on. So what's interesting is most drugs, most natural compounds, we don't know how they get into the brain. They've not been studied. So we don't know, you know, to study them is not easy. You've got to radio tag them, or, you know, and then you've got to follow them. Are you gonna, is a human gonna let you do that? Are you gonna have the capabilities to do that? So you can tell I'm rambling because it's late in the afternoon. I tell you, my brain works best in the morning and the evening, and it's pretty terrible in the afternoon. I don't know if you've noticed this, and your sodium fluctuates that way. It's highest in the morning and in the late afternoon, and that's when people get their migraines most frequently.
Well, I think we need to call it to a close, Mike. You have been utterly fantastic.
Yeah.
And what you're doing, it couldn't be more important. It just still is amazing that at this stage of the game, common brain disorders, the causes of them are not known. But you are beginning and making a huge difference. So thank you so much for being with us today. I think all of us can show our appreciation.
Yeah.
Yes.
My pleasure. Thank you all.
Yes. So next week, we have our convocation at 4 o'clock, and our speaker is Lucia Chappelle, who is a minister with the Metropolitan Community Church. And Metropolitan Community Church is a ministry of the LGBTQ community to the rest of us, to the rest of the world. And it's an international denomination. And Lucia is going to talk about the history of the MCC up till today, and she'll share some of her own experiences as a minister of that denomination. So join us next week at 4 o'clock. And again, Mike, beautiful. Thank you. Thank you so much.
Thank you.
Thank you. And tell us hello.